So today's speaker is Lauren Robinson, who is a sequencing and informatics specialist for the Great Lakes region at Illumina. Lauren is a graduate of Kent State University in Ohio. She majored in biochemistry. After college, Lauren worked as a research assistant at Northeast Ohio Medical University, studying age-related changes in GABAergic neurons related to hearing loss. From there, she moved on to Swift Biosciences, where she led the application support department for all of Swift's next-gen sequencing library prep product lines globally. She took on her current role at Illumina in April of last year, and she told me that she's eager to start meeting folks in person rather than through video conferencing. So hopefully we'll be able to do that soon, but for now, welcome to our Indiana CTSI Access Technology Seminar Series. We're looking forward to your presentation.
Awesome. Thanks for that wonderful introduction, Jill. And thank you. I just wanted to say thank you again to all the core directors for sponsoring this event. Really excited to be here and talk to all of you. So today we're going to talk about the title of the talk here is an introduction to homebrew library prep methods for Illumina sequencing and some critical factors we want to kind of think about here for success. And so some of the methods that I'm going to touch on and some actually complementary methods to these are going to be ChIP-Seq, Ataxic, HiC, and a little bit of information related to Crispr screening. Oops. Okay, there we go. All right, and so our agenda here, we're going to start with some CRISPR related information, specifically CRISPR screening, and then we're going to kind of get into Ataxic, a couple different methods there. Then we're going to talk about ChIP-Seq, cut and run, cut and tag towards the middle of the presentation. And then we're going to follow up with HiC and Capture-C and some newer methods along that space at the end of the presentation. And then I'll open it up for questions at the end. And I just wanted to preface here with the intended audience. So intended audience for this presentation would be folks who may be curious about some of these methods or may also be thinking of designing an experiment with any of these methods being discussed today. All right, and so first, we're going to start talking about some CRISPR screening, and this is definitely one of my favorite applications for sure. I've got a couple CRISPR books I just ordered on Amazon as well, but really the ability to modify the expression of single genes and proteins has become one of the most important tools in molecular and cellular biology. And so several methods such as CRISPR have been developed to allow for specific gene manipulations in cells, which have become known as genome editing. And some common applications of CRISPR technologies can be seen in the figure to the right here. But today we're going to focus on so-called CRISPR screening, which is essentially a really large-scale genetic loss of function approach designed to essentially pick out a few needles in a haystack. And so it allows you to discover key genes that elicit a specific function or phenotype for a cell type. And so I've kind of listed some examples here shown in the text to the left of this slide. And then while also, you're able to start with a pretty broad hypothesis. And in some cases, almost starting with, all of the genes in the genome. And so these CRISPR screens then enable you to generate really a new narrow hypothesis as to what you're able to get from a genome-wide CRISPR screen is really a short list of candidate genes or sequences that produce that physiological response under investigation, whether that be, for example, drug sensitivity or resistance or susceptibility of environmental toxins. And then once you get this list of candidate genes, you can then investigate those further. All right. But before we dive deeper into genome-wide CRISPR screens, I wanted to just kind of quickly review what CRISPR is. And so CRISPR was initially discovered as a prokaryotic adaptive immune system. However, now the system's been adapted into mammalian cells to generate really a simple and efficient means of creating targeted loss of function mutations for one example of its uses. And Cas9 proteins are able to generate precise double-stranded breaks at target loci determined by so-called short guide RNAs or sgRNAs. And in the absence of a homologous template, these breaks are repaired by really a super error-prone non-homologous end joining mechanism, leading to the production of indels. And so when targeting coding regions, knockout libraries can be generated by frame shift mutations resulting from this production of mutations. And so now there are a few key things needed here in order to generate these knockouts. And so the first thing needed here is your nuclease or your Cas9. And this has to be expressed and translocated to the nucleus in order to function properly. And then the second kind of key thing here is your sgRNA, which is actually going to guide and direct Cas9 to a specific site in the genome that you want to cut. And so guide RNAs are typically comprised of a cRNA sequence or a 20 nucleotide target specific sequence that's identical to the genomic target region, a Cas9 binding handle sequence, or essentially where a nuclease Cas9 is going to bind, and then a terminator sequence. And now a prerequisite for binding of the sgRNA to the target site is actually the presence of what's called a PAM motif. And these are typically 2 to 6 base pairs in length. And this is going to actually follow that 20 nucleotide recognition sequence or cRNA sequence. And the PAM motif sequence is actually required for a Cas nuclease to cut. And is generally, again, found about 3 to 4 nucleotides downstream of the cut site. And one thing to note here is that your PAM motif is going to actually vary with the endonuclease being used. All right, so we've reviewed the components of CRISPR. Let's actually move on and kind of start discussing pooled CRISPR screens here specifically. And so the basic idea of CRISPR screening is to knock out every gene that could be important, but to only knock out one gene per target or one gene or target per cell. And so the intended result here is a population of cells with a different gene knocked out in each cell in your dish, essentially. And as a result, some cells are going to die, but others are actually going to survive or even grow better and become the predominant cell type. And so most often CRISPR screens are conducted to identify genes that allow cells to survive under specific conditions, as we mentioned previously, such as drug treatment, or to identify genes that determine sensitivity to a particular drug or treatment. And so most CRISPR-based screens utilize pooled libraries, which are actually going to consist of a pool of sgRNAs or guide RNAs and a lentiviral vector backbone. And so that's going to be used to transfect or transduce in bulk your cell line of interest. So you're really actually using this lentivirus, or in some cases AAV, to help you incorporate the sgRNA sequence into the cell's genome. So the viral genome is actually also going to be inserted here as well. And so generally the first step is to design a pool of oligos that are going to be used to make lentivirus here. And so each oligo in the pool needs to contain the guide RNA sequence and must have sites at each end to allow cloning into lentiviral gene-containing plasmids. And then typically you want at least 3 guides or target sequences per gene. So you can kind of be really confident and make sure that the effect you're observing is actually from the guide-directed knockout and not really a result of the integration of the viral genome into your sample, for example. And then from plasmids, lentiviruses are produced as a pool containing thousands of CRISPR targeting sequences with 1 targeting sequence per virus particle. So you only have really the directions for 1 guide per virus. And then your CRISPR library or your sgRNA library is basically your batch of lentiviruses produced from the pool of oligos. And typically, if you order a library from, let's say, Addgene, for example, it's supplied as a glycerol stock where each plasmid needs to be amplified before use. And you'll need to actually amplify and deliver the plasmids to so-called producer cells or bacterial colonies that are going to actually produce these lentivirus particles for transduction of the target cells. And so one of the most critical parts of a CRISPR screen is the transduction step. And it's important to ensure only 1 virus and thus 1 knockout per cell. So you can kind of be confident that what you're observing is due to that 1 knockout gene. And this is best achieved by what's called a low multiplicity of infection or an MOI of under 30%. And so this is going to mean that an excess of cells will need to be used So at least like 100-fold higher than the number of plasmids. And so as a result, a large portion of cells are actually not going to be transduced in your screen. And then additionally, it's important to infect at least 500 cells per targeting sequence so that you can be sure you're obtaining results within the sensitivity of the assay here, or that, in other words, we're doing air quotes here, maintaining coverage. And then after infection, cells are incubated for a few days to develop that phenotypic CRISPR-mediated change or after, which is silencing of a gene or sequence. And then in the next step, a positive or negative selection is going to be applied. And so in a positive selection screen, a positive selection pressure is actually going to eliminate all cells that don't survive the selection. And so, for example, something like drug sensitivity. And then the few cells that do survive can then be sequenced and their sgRNAs identified and quantified. And then on the other hand, a negative selection screen, most cells are actually going to survive in this case. And so therefore, you're actually going to need to sequence the starting population, then apply your selection pressure or your selection, and then perform another round of sequencing on the surviving cells to identify, a list of sgRNAs that are underrepresented, or essentially, in other words, cells with resistance genes knocked out, for example. All right. So one of the most popular genome-wide CRISPR screens, or one of the most popular libraries, was developed by this group at the Broad, which is called the Gecko Library, or essentially genome scale CRISPR-Cas9 knockout. It's kind of where they get Gecko from. And so the Gecko library of sgRNAs targets 5' constitutive exons of about 18-ish or so thousand genes in the human genome, with about 3 to 4 guide RNAs per gene, or a total of about 65,000 guide RNAs. And so additionally, this group actually designed their lentiviral vector to deliver Cas9, the sgRNA, and actually a pyrimycin selection marker, which allowed them to select for transduced cells. Because on the last slide, we saw that, essentially you're going to have a pretty large fraction of cells that are not going to be transduced. And so that pyrimycin selection marker here allows them to kind of get rid of those cells, so they're not wasting sequencing reads downstream. All right. And so here this group tested the efficacy of their Gecko library by demonstrating its ability to enable both negative and positive screens. And so for their negative selection, they profiled the depletion of sgRNAs targeting essential surviving genes by transducing both human melanoma and stem cell lines with their Gecko library. And here they're actually able to reveal, after deep sequencing, a significant reduction in the diversity of sgRNAs in the surviving cells. And then in order to test for their positive screen efficiency, they look to identify gene knockouts that result in resistance to a BRAF protein kinase inhibitor, PLX, in melanoma, in the same melanoma cell line. And at the end of the 14-day treatment, 14-day PLX treatment, they were able to identify the enrichment of multiple sgRNAs targeting genes that, when knocked out, could contribute to this PLX resistance. And so this includes both the identification of previously reported genes and also, to note a handful of genes not previously reported in PLX resistance. And so they did discover some novel resistance genes with this particular experiment. And then in addition to Gecko, there's actually a couple other groups that have developed libraries of their own, including the Toronto Knockout TKO library generated by the Moffitt Lab, and then also the Brunello libraries from the Broad Institute Genetic Perturbation Platform, or GPP lab. All right, so let's talk about some QC measures here in your screens to really ensure that you're as successful as possible. And so one of the most critical QC steps here to include in your screen is to ensure that your library amplification was successful and that all plasmids were amplified without any being lost, as the bacteria transformed the library grow and replicate. And so essentially meaning, we want to make sure that we're starting our screen with an even representation of all of our guide RNAs. And so the best way to kind of do this is to include a sequencing step for quantification of the plasmids to ensure that all your plasmids have amplified evenly, and to actually also ensure that no plasmids were lost or amplified preferentially, so that you're not missing or overrepresenting any guide RNAs. And so to do this, you're going to extract DNA from colonies and either sequence the entire plasmid sequence with a library prep like Illumina DNA Prep or Nextera XT, or design primers that will target the sgRNA or guide RNA sequences specifically. And so you're going to want to actually ensure after this that you're sequencing to about 100 to 1000x coverage. And so typically it's helpful to sequence both pre and post amplification so that you can compare the numbers before and after. And so then you're going to rank the plasmids by abundance, scale to 1, to create what's called a cumulative wealth distribution curve, or also known as a Lorenz curve. And then by calculating this area under the curve, you can get a sense of the uniformity of the library distribution, with an AUC of 0.5 being basically equivalent to perfect uniformity shown here. But ideally what you want is an equal AUC before and after amplification, as shown in B and C here, so that you can ensure that no guides are lost or over amplified during amplification. Okay, so we've identified a nice QC step. We've talked about CRISPR screens. We really want to at this point kind of determine or perform our hit identification. And so in other words, really, how do we quantify those sgRNAs present after a positive or negative selection step in order to determine the differentially represented knockouts? And so this is actually going to require downstream NGS as a readout. And so there's a few ways to get the pool of knockout cells ready for sequencing on an Illumina instrument. However, one of the most common methods is by designing Pcr primers that are going to recognize the viral vector backbone sequence and that are flanking that sgRNA sequence. And so essentially allowing you to directly sequence that sgRNA, which is shown in yellow here. And so in this paper from the Rana lab at UCSD, they designed a set of Pcr primers able to effectively amplify that constant lentiviral integration sequence, flanking that sgRNA for successful RNA library prep. And so this group actually designed their primers for the Gecko V2 library. As a result, designed the primer binding sequences specific to that lentiviral integration sequence found in this screen. But, you can actually, alter that primer binding sequence to make it compatible with, for example, the Brunello or TKO libraries. And then essentially, you're going to, perform a couple of steps of PCR, and you're going to have those primers again binding to that viral integration sequence. And so their approach utilizes a two-step PCR in which this first PCR utilizes primers that are going to, what we just talked about, recognize that constant lentiviral integration sequence, followed by a second PCR, which is going to be used to add on the Illumina adapters. And so this design utilizes what's called a nested PCR to help kind of increase sensitivity and specificity here. But there's actually some third-party providers like Takara that actually sell library preps for CRISPR designs, actually utilizing only the second PCR here. So ultimately, this step is going to be up to you and your lab to kind of validate. But many folks I see, definitely do both of these methods. And one thing to note here, though, is that you really want to make sure that your primer binding sites are staggered. So these sequences that are binding to that lentivirus, in order to kind of help increase color balance and diversity for sequencing, because you're going to actually start each read with the same lentivirus sequence, really otherwise. And, you may also want to consider spiking in some phi-X here as well, with different, phi-X spike-ins per instrument. And some other workflow designs also include the addition of, let's say, like a custom sequencing primer to start the read one sequencing at that actual guide sequence, for example. So your custom sequencing primer binds to this lentiviral sequence, so you're missing that low diversity portion altogether. But again, kind of what you decide to go with here, is definitely going to be up to your lab and some validation, but there are some options to kind of get over that low diversity there. And then now once you kind of get your second PCR product here, we are ready for sequencing. We've got our adapters on. And typically you want a read depth of anywhere from 100x to 1000x per guide RNA sequence, depending on the type of screen you're doing. And so negative screens are going to require more depth or more sequencing depth than positive screens. And so, for example, if you have a library with 60,000 guide RNAs, so kind of similar to that Gecko V1, you're going to want to sequence to at least 6 million reads per sample. All righty. Now, in addition to bulk approaches, we're taking basically our bulk pool of cells, extracting that DNA and sequencing to count those guide RNAs. There's also some single cell approaches that allow us to kind of take this a step further. And so with a single cell approach, you can actually identify what particular guide RNA is expressed in any one given cell. And as a result, you can actually also see the full transcriptional profile of that particular cell as well. And so this allows you to not only identify that perturbation, but to also look at how this particular knockout is affecting the global gene expression phenotype of that particular cell, which could really help us better understand why our particular knockout is behaving a certain way. And so whether that be, again, resistance or sensitivity to our experimental conditions. All right. And then one of the most popular methods of combining single-cell RNA-seq and sgRNA detection is a method by the name of Perturb-seq. And so this combines droplet-based single-cell RNA-seq with a strategy for barcoding CRISPR-mediated perturbations to allow many perturbations to be profiled together, making it compatible with some of these large screens. And so for the sake of time, I'm not going to go through all of the details of how this is done here, but actually 10x Genomics has some product offerings in this space that allow you to combine single-cell RNA-seq with feature barcoding technology to actually detect sgRNAs within the same cell you're performing RNA-seq. And one thing I want to note here, though, is that this workflow requires the integration of what's called like a capture sequence in your guide RNA. And so 10x is saying either at the 3' or 5' portion of this sgRNA. And so you're going to need that for compatibility with their feature barcoding, and more information could actually be found on their website there, but happy to answer any questions afterwards. And then overall, I guess I just wanted to stress the value here of this method, which really is like a means to analyze the transcriptional effect or phenotype in addition to the survivability of a particular knockout. It could actually potentially lead to a decrease in the cost and amount of time needed for basically to further assay, those complex effects of these perturbations or knockouts that you're observing in your screen. Okay. So let's shift gears now to our next topic, which is going to be ATAC-Seq, or sequencing regions of open chromatin. And, I'm going to start with this question, how do we sequence openness? And actually the answer here is pretty clever. So as we know, DNA is packed into the nucleus through a hierarchy of folding around histone proteins to form nucleosomes, and then the compaction of nucleosomes into chromatin. And so this hierarchical system of packaging isolates inactive regions of the genome and leaves biologically active regions such as promoters, enhancers, and other regulatory elements accessible to the transcriptional machinery. And so at a high level, ATAC-Seq is a method that allows us to profile these regions of closed or open chromatin, or to sequence these regions accessible to these regulatory elements, regardless of the mechanisms responsible. So for example, histone modifications, etc. All right. And so why is chromatin accessibility important? Well, it's actually one of the critical necessities for anything meaningful to begin. And so the cell needs to compact DNA to fit into the nucleus, as we discussed on the last slide, and about 97% is packed into so-called heterochromatin, so quiet and dormant. But as different parts of the genome are needed, such as when cells differentiate or respond to stimulus, this chromatin needs to unfold to become accessible enough for transcription factors to kind of get things started, and for chromatin to open up into what's known as euchromatin, the active 3% of our DNA. And so the function or dysfunction of this process can actually be pretty impactful genome-wide. And it's important to distinguish that ATAC-Seq is often done with RNA-seq, but it's really not the same. And so ATAC-Seq doesn't tell you what is expressed, but tells you the why the why not something's being expressed, or actually what could be potentially expressed soon. So it's a really nice complementary method to RNA-seq. All right. Okay. And so the Ataxic method relies on next generation sequencing, library construction, using the transposase TN5 enzyme. And so NGS adapters are loaded onto the transposase, which allows for simultaneous fragmentation of chromatin and then integration of those adapters into open chromatin regions. And then the library that's generated can actually be sequenced by NGS. And then the regions of the genome with open or accessible chromatin are mapped back to the reference. And so the main advantage of Ataxic compared to other techniques like FAIR-seq or DNA-seq, for example, that investigate similar chromatin features, are the lower number of cells that are required for the assay and the relative simplicity of it's really two-step protocol. And you're going to start with fresh or frozen cells. So depending on the protocol, as little as 500 can be used. And then the nuclei are all that is needed. So those are actually spun out. And then the nuclei are exposed to that TN5 enzyme and buffer, which are actually reagents. I should note we sell at Illumina. And enzymes like TN5 actively avoid protein blocks, including nucleosomes and transcription factors. And they're actually only going to chew up and create libraries from accessible regions, as you can see shown here. And then really you've got some basic purification and an indexing PCR. And really that's kind of it, you're done. It's about a 2 to 3 hour protocol, I would say, for an experienced user. And then once sequenced, again, reads are going to stack up similar to RNA-seq, and that you can count the reads mapped to a loci. But here, instead of counts relating to transcripts detected, it relates to how open or accessible that region of the genome is. And so here, deeper coverage or more open equals more open, and lower coverage means more closed. All right. And then what you should all know is that in the years following actually the release of the original Ataxic method, the assay's actually gotten better, largely due to the work from the original publishing labs and others with what's called the OmniATAC protocol. And so the major improvement in the new workflow includes 3 major things. And so the first being the use of multiple detergents such as NP40, tween, and digitonin to improve the permeabilization across a wide array of cell types and to remove mitochondria actually from the transposase reaction. And then the second includes the post-lysis wash step using tween to kind of further remove mitochondria and to increase the complexity of the library. And then third and lastly, the use of actually PBS in the transposase reaction to increase the signal to background ratio. And, a lot of these methods or a lot of these alterations, getting rid of some of those mitochondrial reads, again, kind of helps you decrease your sequencing depth as well. And so as a result, the new method requires less input cells, as low as 500 compared to tens of thousands previously. And it also opens the door to frozen tissues as well. And like I just mentioned, the sequencing the sequencing to noise ratio is significantly improved here as well. There's also a significant reduction in sequencing costs as the new method is able to significantly reduce reads that, as I mentioned, map to mitochondrial DNA, and then an overall higher percentage of the reads that map to those peaks of chromatin accessibility we're really after. Okay. And so another approach folks are using more commonly these days is single-cell Ataxic. And this actually uses that same TN5 transposase digestion to look at the accessibility of DNA, similar really to what's done in bulk Ataxic experiments, only at single-cell resolution here. And so similar to bulk ATAC, the data comes out as peaks corresponding to regions of open chromatin with more coverage, meaning more openness. And then by looking at these particular peaks from the data, we can actually look at a couple things here. So one, we can perform cell state classifications. And so we can actually cluster cell types based on their epigenetic profiles. So regions of open chromatin rather than their expression profiles. So now we're actually capturing that what could be expressed in addition to that what is currently being expressed with single cell RNA-seq. And then additionally, we can also infer which genes are being expressed at single cell resolution. And so demonstrating by, again, regions of open chromatin, while at the same time including information regarding transcription factors. And so because in this case, we're getting the DNA sequence, we're able to actually determine the footprint of potential DNA binding motifs underneath these open areas where transcription factors could actually bind. And so further expanding on the information we're able to achieve with this approach when compared to, just single cell RNA-seq alone. And then 10x Genomics actually has a pretty nice kit for running single cell Ataxic. And they actually also released sometime last year their multiome kit, which can actually perform both single cell Ataxic and single cell RNA-seq within the same cell, really allowing you to kind of get both expression and chromatin confirmation within the same sample. And this method is actually previously referred to as snare-seq. Alrighty. And so finally, before we're going to move on to the next section, I just wanted to talk a little bit about these sequencing requirements for Ataxic. And this is kind of going to vary depending on our application. And I should note too that these are all, starting recommendations, and you may kind of need to play around with these as it pertains to your particular sample types. But first and foremost, though, I wanted to just kind of stress that we want to use paired end reads here. Because they're actually going to offer a number of pretty significant benefits to us. And so firstly, we're going to increase our alignment rates of unique fragments. And actually, additionally, both ends are going to actually tell us the insertion point of that transposase. So it's really going to actually double our data when you sequence from both ends. And so in addition to this, actually paired end reads can also allow us to determine the size of our fragments as well. And actually this fragment size can then be used to identify, nucleosome positions as well as nucleosome free regions at these relatively accessible regions of the genome. And so in terms of read depth here, for identification of differentially accessible regions, you might want to start with at least 50 million paired end reads to get started or greater. And then when you're wanting to kind of take a deeper look and pull out potential transcription factor footprinting or potential binding motifs, you might want to have a little bit greater read depth here, maybe starting recommendation of 200 million paired end reads. And then lastly, for these single cell experiments, you're going to want to start with at least 50,000 reads per nuclei or per cell. Alrighty. Okay, so we've gone through CRISPR and Ataxic. Let's kind of move on to our next topic, which is discussing methods that are going to help us detect some DNA protein interactions, for example, like ChIP-Seq. All right, so really, starting off here, let's talk about what ChIP-Seq can tell us. It's an excellent method of mapping protein DNA interactions, including transcription factors, as well as histone modifications. So it can actually be used to answer a multitude of questions involving the interactions of DNA and proteins. And so, for example, it can be vital in understanding why, let's say, some genes may be turned on or off, or also, up or down regulated. And so that whether that be on, let's say, the transcription factor side, assessing the binding site of either repressors or activators, or on the histone side, such as, certain acetylation or methylation groups. And then, these alterations to our histones can actually also determine either open or closed chromatin. And so there's very few papers in the field of actual transcription, gene regulation and epigenetics that actually, don't contain at least, let's say, one ChIP-Seq experiment. And we talked previously about how Ataxic can help us determine possible binding motifs for transcription factors or these regions of open or closed chromatin. So let's say, adding ChIP-Seq or a ChIP-Seq experiment here could give you an additional level of insight regarding binding motifs for a specific transcription factor, as well as some of those histone modifications that could lead to regions of either heterochromatin or euchromatin. All right. All right, so we know why ChIP-Seq is important, but how is it done? And so the basis of chromatin immunoprecipitation is to fix DNA to proteins to really freeze these interactions in time, and then to extract the DNA, shear it to workable fragment sizes, and then to enrich for fragments of interest by immunoprecipitation. With antibodies that are actually going to be specific for DNA binding proteins, for example, like transcription factors or those histone modifications. It really kind of just depends on what we're interested in. And then the immune enriched DNA fragments are then able to be identified and quantified using next generation sequencing. And so after sequencing, reads near the protein or modification of interest will actually be mapped back to the genome, and you're going to actually use these peaks to infer where your targets of interest are located across the genome. So we just kind of had a high level here. Let's kind of dive more specifically into the details here for standard ChIP-Seq workflows. And so first, cells are fixed in order to kind of cross-link these proteins to DNA using like formaldehyde-based reagents, for example. And one thing to note here is actually that overfixation of chromatin can actually lead to a reduced efficiency of fragmentation. It can actually inhibit the binding of some antibodies to their target. So that's just one thing to kind of keep a note of there. And then next, you're going to go ahead and lyse your cells and move on to chromatin fragmentation, which is actually essential for a successful experiment and may actually determine the resolution of your assay as well. And so there's kind of two common methods for fragmentation, one including enzymatic digestion using micrococcal nuclease or MNAs, which cleaves double-stranded DNA between nucleosomes, or another via sonication, which actually uses mechanical forces to fragment chromatin between and within nucleosomes. And then you should probably check your fragment sizes here after fragmentation as well before moving on to immunoprecipitation. And so then the next step here includes antibody binding. And I want to note that choosing your antibody to use in a ChIP-Seq experiment is probably the most vital part of the entire process, because that specificity of the antibody and the degree of enrichment achieved in the affinity precipitation step can really honestly make or break your experiment. And so you're going to want to make sure you're using ChIP-Seq validated antibodies here. And so after antibodies are used to capture your protein of interest and its bound DNA, these complexes are then going to actually be affinity captured on an antibody binding resin, which is actually going to be composed of a magnet and an agarose beads conjugated to protein A, which is actually going to bind to that IgG portion of your antibodies. And then beads are typically incubated here for about 2 to 4 hours. And after this, you're going to have some wash steps to remove these non-antibody bound DNA, followed by, eluting your targets of interest from the protein A beads using a detergent and heat. And then next, you're actually going to need to remove the chromatin crosslinks from fixation using high heat and high salt. And proteinase K is added to actually digest the proteins and added antibodies, so that all we're left with at this point is DNA. And so lastly, you're going to purify your DNA, and you're actually going to proceed with just a standard double-stranded library prep kit to add on your sequencing adapters. All right, now regarding sequencing requirements for ChIP-Seq experiments, depending on what you're looking at, you're going to need to adjust your read depth. And so, for example, for transcription factor ChIP-Seq, the ENCODE project recommends that each replicate should have about at least 20 million reads. However, if you're looking to do histone ChIP-Seq, you may need to increase this read depth to at least 45 million reads if you're looking to identify broad peaks. And more details can actually be found on either Roadmap Epigenetics or ENCODE for specific guidelines to follow. You can see here a couple examples of what's considered narrow versus broad peaks. All right. Okay, so, although XChIP or, ChIP-Seq in a traditional sense has been around for a long time, it can actually potentially produce poor resolution, suboptimal signal to noise, and actually false positives due to the chemicals used to cross-link chromatin, the strong forces used to fragment it by sonication, and the detergents that are used to make these fragments soluble. And so a more recent method developed by the Hennekoff lab at the Fred Hutchinson Cancer Center called cut and run, or cleavage under targets and release using nuclease, can actually overcome some of these limitations associated with traditional ChIP. And so cut and run does not actually require fixation. It actually uses a protein A MNase fusion protein that's actually going to guide the cutting of the MNase enzyme to the protein of interest, while that antibody is still bound to intact chromatin after the addition of calcium in this case. And so this means that nearby DNA is actually going to be cut into small fragments. And because of their sizes, these fragments can actually then float out of the nucleus. So actually here, leaving that rest of the uncut genome behind. And because of this, cut and run is actually can actually significantly reduce the background noise when compared to traditional ChIP. And as a result, this is the big one here, requires about 1/10 of the sequencing depth and is compatible with much lower numbers of starting cells. And so one thing I want to note here is that cut and run produces fragments of about roughly 100 base pairs for transcription factor complexes and about 150-ish for histone mapping. And these are pretty small fragments. And because of this, it's really important to choose a downstream library prep kit for NGS that can actually handle these smaller fragment sizes, and to kind of keep these sizes in mind during your size selection steps, because you don't actually want to accidentally get rid of those smaller fragments. And then the Hennekoff lab here used just like a standard double-stranded library prep kit and ensured that, they adjusted those spry ratios so that they could kind of keep everything 100, 150 base pairs and above. And then they also performed a right-sided cleanup or Ampere bead cleanup to kind of get rid of some of the larger fragments that may have been left over. And then another way they kind of tried to get rid of some of these larger fragments was by increasing the number of PCR cycles used in their final library amplification to kind of help them favor the amplification of these shorter fragments, because ultimately these shorter fragments are the ones that we're going to want to sequence here. All right. So as you can see on this slide, you're able to really significantly reduce the background noise observed with cut and run when compared to traditional X-ChIP. And as shown by the ENCODE data on the slide, you can really kind of see this. Additionally, cut and run is able to support as low as about 100 cells for histone modifications, as shown in the figure to the left, and as few as 1,000 cells when looking for CTCF peaks or transcription factors, as shown on the figure to the right here. In addition to the lower cell input with cut and run, base pair resolution can actually be achieved here with as few as 5 to 10 million paired end reads per sample, so significantly less read depth here when compared to traditional ChIP. And paired end sequencing is also, again, recommended here because single end sequencing can actually sacrifice some of this resolution and discrimination between transcription factors and these neighboring nucleosomes. Okay. In addition to cut and run, there's also another pretty clever method that came out of the Hennekoff lab as well, called cut and tag. And there are also a ton of major similarities here between cut and tag and cut and run. And really the only major difference here is that instead of MNAs guiding that cutting around that bound antibody within the cell's native environment, we're actually instead using a TN5 transposase, in this case, loaded with sequencing adapters. So just like with cut and run, where we have our TN5 fused with protein A, which is gonna, again, bind to that IgG portion of our antibody, here we have that TN5 directing that antibody-guided tag mutation on either side, and you can kind of see that right here. And now because, in this case, we have that TN5, we're also going to be adding these anchor portions of our adapter shown in red at this step as well. And so this really allows us to kind of almost have almost a finished library for sequencing after purifying our DNA. And so all we really need to do here is a single simple indexing PCR step, and essentially we're done. It's pretty simple. That's pretty much it. And it's a much quicker workflow when compared to cut and run. And the entire process can be actually completed in a single day. And so now, in addition to saving us time, the tag mutation step can also make it compatible with inputs down to a single cell, as we no longer need to worry about the input limit of many ligation-based library preps out there. And instead, we just kind of need to worry about amplifying up those anchor portions here of our adapters with PCR. And because this method can support single cells, it can actually also be great for heterogeneous populations, such as blood, brain, pancreas, or liver. All right, I did talk up cut and run and cut and tag quite a bit, but in some cases, new is not always better. And so for those of you who watch How I Met Your Mother, I'm going to have to disagree with Barney on this one. But before we move on to the next section, I wanted to just take a moment to highlight that although cut and run and cut and tag can offer a lot of advantages when compared to traditional ChIP-Seq, there's still some cases in which standard ChIP-Seq may be the best way to go for your experiment. And so, for example, many transcription factors are not abundantly expressed, weakly or transiently bind to DNA, or actually indirectly bind to chromatin. And so for these cases, chromatin cross-linking and sonication via these traditional methods are probably a mandatory step to detect the protein-DNA interactions. And additionally, many ChIP antibodies are validated to work with fixed samples and may not work well with native conditions that are seen in both cut and run and cut and tag because the protein epitope availability and cross-link conditions can be a tad different than the native conditions. And so therefore switching from standard ChIP to cut and tag or cut and run would actually need a thorough antibody validation to be certain of the antibody specificity and sensitivity under unfixed conditions. And then lastly, specifically for cut and tag, the TN5 transposase used in cut and tag, has a high affinity to open chromatin regions, as we saw with the Ataxic. So it may be preferentially suited for analyzing histone modifications or transcription factors associated with actively transcribed regions of the genome. And maybe not so much for the analysis of regions of the genome that are silenced or contain heterochromatin. All right, so let's move on to our last section in this webinar, which is going to be methods to help us detect some long-range DNA interactions through methods such as Hi-C and Capture-C. All right, so before we begin, let's just have a quick review of the importance of some long-range interactions here, such as enhancer-promoter communication. And so roughly, about 13% of the human genome is likely devoted to enhancer function as defined by ENCODE. And so how do these enhancers target to specific genes from a distance? And so very often these enhancers can be actually looped to the promoters of the gene. And it's this process that actually got folks interested in how chromatin is looped with each other and interacts in order to start transcription. All right, now one of the most popular methods to study 3D chromosomal interactions is Hi-C. But before we get into the specifics of this method, let's kind of first review the organization of our DNA in the nucleus using these Hi-C contact maps on the top and the animations of the actual chromatin features on the bottom to kind of help guide us in our discussion here. And so you can imagine that filling a human genome into a nucleus is kind of similar to filling a string that's about 44 miles long into a basketball. And so how do you package it so that the correct interactions can happen? And so mammalian genomes are folded at multiple scales, and each scale kind of highlights an important level of organization and function. And so first, individual chromosomes are actually organized in distinct nuclear territories, as shown here on the far left, with each chromosome representing a single color. And as you can see, chromosomes are pretty isolated within their own territories, suggesting a higher degree of intra versus interchromosomal interactions. And this can be observed here by this contact map. And then the next level down in resolution within a chromosome are specific compartments or AB compartments that separate euchromatic and heterochromatic regions. And so A compartments shown in red overlap with transcriptionally active regions, where blue or B regions shown in blue correspond with regions of closed or inactive chromatin. And at the sub-megabase level within these compartments, an additional layer of chromatin folding can be observed with so-called topologically associated domains, or TADs, which are defined as genomic regions with increased contact frequency. And so these are shown as those loop-looking structures that are kind of pinched together here on this figure, and they can be represented by these triangles on the contact map. And then TADs are also closely related to another prominent chromatin feature, which are these loops. And loops are really actually that focal point of interaction within a TAD. And you can kind of see these generally as like that top of the triangle here. And many of these chromatin loops are bound by cohesin subunits like RAD21, SMC3, and CTCF, which actually CTCF usually displays the convergent orientation here as well. And you can see this shown on the figure to the far right with these little arrows corresponding to CTCF. Okay, so, we know how and we understand, how our DNA is strategically organized in the nucleus, but how do we capture that information with sequencing? And so one of the most popular methods here is HiC, which is based on the original 3C or chromosomal conformation capture method. And so how does HiC capture this 3D information? So in order to explain how HiC captures the 3D information, it's important to think of it in the context of a standard library prep here, which starts with our cells and tissues, and then we fragment this DNA and add on our adapters and amplify with PCR. But, in step one, that fragmentation step, we're actually losing this critical spatial proximity information, and we're only preserving the sequence. However, with HiC, we're actually wanting to preserve both sequence and structure. So the extraction and upstream processes need to be a little bit different here in order to preserve the structure of these long-range interactions. And so with HiC, you're going to begin with your cells. And the first step here is to cross-link these cells because we want to actually freeze these long-range interactions in time with formaldehyde. And so then you're going to actually take this cross-linked DNA and digest it with restriction enzymes, and then add, and then those ends here are actually going to be, that were digested, filled in with biotinylated nucleotides, and they're actually going to be ligated together. And so what's kind of interesting here is that the orange and blue become ligated into one piece, where before they were actually not joined in any way whatsoever. They were just kind of held together by proteins. And so here you've actually created this artificial fragment, and this ligation junction here is that piece of DNA that captures our contact information. So this is kind of ultimately what we're going to want to sequence. So we want to sequence all the blue-orange chimeras. And so next, you're actually going to reverse those cross-links and get rid of those proteins. And then you're going to purify and fragment your DNA. And so we're then going to actually use these biotin markers, so those little purple dots shown on our orange-blue chimeric fragments, to actually enrich for our ligation junctions, or fragments, again, containing both blue and orange. And we're going to do this with streptavidin beads and to actually leave the only blue and these only orange fragments behind, because they don't have really any of this information after, and we're just going to waste some sequencing reads on those guys. And so finally, we're going to proceed to standard, basically a standard double-stranded library prep with our final chimeric fragment containing blue and orange. And so we're going to add our adapters on here, PCR amplify up for sequencing of our chimeric library. And one important thing to note here is that you absolutely, I cannot stress this enough, need paired end reads here for sequencing. And this is because we need to capture the information from both ends of our library molecule so that we can actually detect both the orange and the blue guy and map this interaction back to the genome. Now, in terms of resolution power, Hi-C typically can detect down to these chromatin loop structures we discussed previously. However, it's important to note that actually the resolution of your Hi-C data is actually highly dependent on really two major things here. And so the first is the restriction enzymes used. As you know, the more sites that are cut here in this example, or the smaller the fragments, the better really the better here. And actually a lot of many new methods use 4 cutters as opposed to, let's say, 6 cutters used in the past. And then the second thing here is actually the sequencing depth. So first major thing is the restriction enzyme, and second is sequencing depth. And so, for example, if you're looking at chromatin loops or these actual regions of legitimate contact, you're gonna actually need a pretty decent sequencing depth. And again, this is just kind of really a suggestion. This is going to depend on sample types, but you want to think about at least 600 million reads, or if not many more, in order to detect these loops. And then another thing to note here is that most standard Hi-C workflows typically require millions of cells as input. However, there's some methods like low-C or tag Hi-C that enable much lower resolution, down to hundreds or even a couple thousand cells. And additionally, there's also some single cell methods circulating out there as well for Hi-C. Now, Hi-C is great if you're looking to pick up the 3D interactions across the entire genome. However, what if you only wanted to target a subset here? Well, this is kind of where methods like Capture-C come in. And so you can kind of think of Hi-C in the same way you would whole genome sequencing, and Capture-C as like kind of how you would think of whole exome sequencing, is it's only capturing kind of a subset of the genome you're interested in. And so with Capture-C, you're going to actually start out very similar to how you would for Hi-C, but after we add our adapters onto our chimeric fragment and finish our library prep, we're going to follow up with a downstream hybridization capture reaction with target-specific probes that are actually going to pull down the regions of the genome we're most interested in, such as specific enhancers or promoters or genes. You could tile it, across areas for discovery even. And so when we ended up, what we end up here is actually a map of chromatin interactions in only these areas we want. And so the sequencing requirement here is significantly less than what you would need for standard or full genome Hi-C, because we've just basically decreased our target region of interest, which can also help us even increase our sensitivity at much lower sequencing costs, to even hundreds of bases in this case. And for the sake of time, I've got one more slide here, but I'm not going to spend too much time here because we're kind of coming up at the top of the hour. But this is a kind of an interesting paper that came out in 2020, basically using tagmentation to kind of help increase the sensitivity of Hi-C. And so here you would actually be using the TN5 transposase to actually fragment your ligated fragments here. And then you've kind of already got partially ready basically libraries for sequencing. And then you just kind of do that indexing PCR here and you're done. So it is much more sensitive, and you can do about 200 to 1,000 cells, they quote. And I've got the citation here if you're interested in looking into this further. And then to wrap it up, we've got a lot of really great resources on our webpage. We've got a couple of these for all you seq posters, one for DNA, one for RNA, one for single cell applications. So we do have quite a bit of information on our website to help hopefully answer some further questions here. And I wanted to just kind of stop here and just, again, thank everybody for allowing us to speak to you here, and to really kind of have a shout out to the core directors. And again, we're definitely here for you all. And, if you have any questions or any, read depth recommendations, want to inquire about any of these methods for an upcoming experiment, I really encourage you to reach out to your core directors to have a discussion. And with that, I'll open it up for questions.
Great. Thank you, Lauren. It's very informative, very in-depth. We have one question in the chat. It is the read depth that you mentioned is relative to the genome size. When you recommend a certain sequencing depth, for example, 50 million or 200 million for Ataxic, what's the genome size you are referring to?
That's human. So that's for read depth for human genomes or equivalent genome size. If you're looking at something like, for example, yeast, it's going to be way, way less. So it is highly dependent, all of those read depths on genome size, but pretty much all of the recommendations I've given are for human genomes. But again, if you're looking at smaller model organisms like yeast, you'll probably be in the hundreds of thousands of reads rather than millions.
And so does Illumina have kits that are optimized for genomes with high GC content?
Oh man, that's a good question. I would say PCR free is the way to go there. High GC content is not your friend with PCR. So anytime you can kind of do a PCR free method, that's probably the way to go. You're gonna get some more even representation of the entire genome there. So we've got a new PCR free kit. It's called the Illumina DNA PCR free. And I would probably recommend that depending on, how much sample you've got to start with. That's probably going to be a good one to do for high GC rich genomes.
And then what about high AT genomes like Plasmodium?
Yeah, I partially want to say, again, we're not running into the same things there, but I would say probably any highly biased, genomes, whether AT, GC rich, I would say it might be best to consider maybe PCR free, but we can kind of follow that discussion up offline if that's helpful as well.
No more questions at the chat, but if someone wants to raise their hand or unmute themselves, they could go ahead and ask that. Well, if there's no further questions, thank you again. People will have the recording so they can review the information. They have your slides and they have your contact info, so they should be able to get a hold of you. So thank you again. And thank you everyone for attending.
Yeah, thanks for having us too.